We have basically a new website that, let's say, exclusively devoted to this program. So the IPC Community Seminars, which is a very long name there. This is basically hosted on a GitHub. So the all credit for that goes to Abel Carreras, who dedicated his time to build it up. And there you can find all the information on the, let's say, forthcoming talks, previous talks with all the links to the YouTube channel. If you miss talk, basically you can see there, and all these, let's say, tax and related information. So I think this is very interesting space, especially will be interesting in the next years when this program will grow and grow. And related to that, also there's a basic aspect that having, let's say, talking with Gesa, with David, with Ricardo, we have this basically concern that we have a low, let's say, participation of PhD students in this program. And this is something that probably is very important to encourage them, especially if this program basically started right now. We have a sixth talk. We have 90 remaining. So I think this is the program for the next 4-5 years. So this is very good to basically encourage the early stage students to participate and to open, let's say, mind for the different disciplines, especially if the IPC community grows in so different directions like quantum, nano, cosmos, and life. So we will have here basically talks on the whole disciplines and on the time scale and landscape. So I think this is a very good opportunity to see different, let's say, topics. But without much ado, let's go to the star of today. So Fabienne Barroso is our today's speaker. Fabienne is a Ikerbasque professor since 2016 in DIPC, physically located in Material Physics Center. She's experimentalist working on the synthesis of polymers and characterization, especially in the context of development of new materials, nanomaterials. Fabienne defended, got the degree in PhD in chemistry in 99 or 4 at University of Havana. Later,
Chile.
PhD. I'm speaking on the degree of chemistry.
Oh, degree. Okay.
Yes. So University of Havana. Later, PhD 2004, University of Chile, and later postdoctoral stays in Germany at the University of Saarland, and then in Madrid, Instituto de Ciencia y Tecnología de Polímeros, and since 2008 here in the IPC. So I said before, she's an Ikerbasque professor since 2016, working mostly on the polymers, and today's talk probably will be most focused on the special types of polymers, which is a cyclic polymers. And let me now switch to the PowerPoint. So the floor is yours.
Okay, thank you, Marek. I would say good morning instead of good afternoon to everybody. So as Marek said, I will talk about the cyclic polymers. So this, I have been developing my research here within the Polymers of Matter group, where I have many collaborators there, many publications, with many investigators there. I also would like to thank people who I am recently working with, Marek and Luca, who is also here, working in a recent project on cyclic polymers. In Polymath, Estivalis, who is performing a Malditov spectrometry to our samples. Scott Grayson in Tulane University, he's also doing research on our samples, performing mostly Malditov spectrometry. And Jürgen Elgeyer, who is providing me with the cyclic polymers for some specific research. So this is the group. This is a picture in 2019. We have a more recent picture, but I don't have it here. But this is a good picture because I have mostly all my collaborators, students, all PhD students and postdocs here. So Jordan, Ali, and Danny, they were working on these cyclic polymers in my project. Faria was a visitor from Tulane University, working in the group of Scott Grayson. She was also here visiting me. And this is my current PhD student, Andrea, who is also here, working also in cyclic polymers. So this is a polymers of matter groups, and most of the collaborators in the group are who I am working with, they are here. So what a cyclic polymer is? It's nothing else than the polymer that has no end groups. It's a cyclic polymer, as the name is saying. Interesting thing on cyclic polymers is that they were already discovered in the 60s, where cyclic DNA was found by researchers. And then researchers say, why nature synthesize these kind of molecules? So interesting. And in fact, there is not only cyclic DNA, also RNA and other molecules within our bodies, and they dictate most of the participation in our metabolism, etc. So after these discoveries, synthetic polymerists say, okay, let's synthesize cyclic polymers and let's study them, their physical properties and biological properties. And then there is a huge community working on, well, it's not so huge. I would say it's a medium community working on cyclic polymers. If we look at this naive picture with the cyclic rubber and these cables, we can see obvious differences between cyclic conformations and the linear ones. It's the presence of end groups, entanglements. So how they distribute, if they can interpenetrate or not. If these cycles are very small ones, they cannot interpenetrate, of course. And all these characteristics will have an influence on the properties of the polymers. So let's go to the properties. This is a typical physical property that we observe in cyclic polymers, which is a glass transition temperature. Cyclic polymers in the low molecular weight range present higher glass transition temperatures than the linear ones. Then at high molecular weight, they all go to the same temperature, which is called the infinite glass transition temperature. So, and this is due to the lack of plasticizing effect of end groups in the physical properties on the mobility of these polymer chains in the low molecular weight range. The longer the molecular chain, these end groups are diluted, and of course they have no more effects on the glass transition temperature. There is another property, which is the crystallization. Researchers have found faster crystallization kinetics on these cyclic polymers. In this case, it's a cyclic polycaprolactones, and the researchers have found that they explain these experiments because these results because of the reduced viscosity of the cyclic polymers in the bulk phase, which increases the diffusion rates and favor the crystallization of these cyclic polymers. There are also other physical properties. As for example, the rheology is different, they behave different. There is they don't show rubbery plateau and faster terminal relaxation, which is related to the lack of entanglements in the short chains, etc. So in solution, they also have different characteristics in comparison with the linear ones, which is related with their size. For example, hydrodynamic volume. They have smaller hydrodynamic volume in comparison to the linear ones, smaller radius of gyration, smaller solution viscosity. The ratio of these properties, cyclic against the linear one, is this about 0.6, 0.8. These characteristics allows us to characterize the cyclic polymers by size exclusion chromatography or jet emission chromatography. If they have same molecular weight, they will retain later the chromatogram. And this allows to say, okay, we got the cyclic polymer after the synthesis. And this happened at different molecular weight. This is a result obtained by Ali some years ago with some kind of polymer. And so I presented here as an example of hydrodynamic volume. Thermoresponsiveness. There are some polymers, like, for example, Nipan, which is thermoresponsive. That means that when you heat the solution, a water solution of this polymer, it starts to become cloudy. It means that it doesn't like anymore the water. And in the case of this, in this study, the cyclic Nipan, for example, also showed that the temperature at which this process occurred was higher than in the linear one. So this is a heating temperature, this is transmittance. At the beginning, at a low temperature, it is completely soluble, and then it starts to become cloudy. The researchers say, okay, they explain these results as the difference in this process, the dehydration process, and the formation of the coil to globe collapse, and so on. Micelles, for example, Scott Grayson, he synthesized micelles obtain micelles from copolymers or with a hydrophilic and hydrophobic part in the chain. He compared the cyclic polymer with the linear one, and he observed smaller sizes in the cyclic micelles, and also differences in the degradation profile. The degradation profile, this is a molecular weight, how it's reduced as a function of time. He did an acidic cleavage of the polymer, and he observed that it was retarded in comparison with the linear one. So there are many properties that are related to just this cyclic topology, and that we can use it in the future, probably for some applications. This is very interesting, the polymer brushes with the cyclic polymer brushes. This study, these and many studies have been done in the Benedis group in Switzerland, where they have created surfaces, they have modified surfaces with cyclic polymers. By AFM, they studied how these two surfaces modified with cyclic brushes under friction forces behave. And they found lower friction forces in the cyclic polymer functionalized surfaces. And this is, they explained that it's because these cyclic brushes cannot interdigitate, they cannot interpenetrate, and that's why their friction forces are slower. Also they measure the repulsion against compression. They compress the surfaces, and they say, okay, our cyclic modified surface shows higher repulsion. Why? Because they observe higher the density of these cyclic surfaces, polymer on surfaces was higher than in the linear ones. So there are many explanations, and it's just thinking on the configuration, on the conformation or configuration of these cyclic structures. And also they also measure in some nanoparticles modified with a cyclic polymer how it behaves in the presence of protein, and the cyclic brushes avoid the formation of protein corona, for example. So these examples show you the importance of the cyclic polymer in general in the polymer field where I am working in. So let's see how we synthesize these polymers, because this is not a very simple task. This is one of the approaches, the ring closure. We have a cyclic a linear polymer. We can form we can synthesize any linear polymer with any composition. We modify, for example, the in groups, and we can form the cyclic polymers by combining with a linker, or by making the reaction between the streams of the polymer. It can be the same group, different group. Problems here is that we have to work in high diluted conditions. because the end groups have to find each other, and this is entropically very difficult. So, and also to avoid that two linear chains, for example, find each other and react between them. So what we want is that the end groups react against each other. And this is limited by diffusion and by the kinetics. So I bring you some examples of chemical reactions done in this sense to obtain the cyclic polymer. For example, this first work done by Grzelczak in 2006 demonstrated the use of the click chemistry reactions in the generation of cyclic polystyrene, for example. Why this was a very important result? Before that, people used organic reactions that they were probably not too fast and created a lot of side reaction, byproducts, and so on. With this, with the implementation of the click chemistry on the cyclization, polymerization, and the cyclic polymers, he demonstrated that high purity of cyclic polymer could be obtained. This is why it's called click. Well, it's a name provided by the organic chemists, but it means that it's a very fast reaction. And this is one of the typical click reaction. This is a copper, a catalyzed acide, alkyne, cycle addition. This is the reaction with this alkyne, this acide, create this, form this triazole. The good thing of this is that you can synthesize the linear polymer by any polymerization technique. In this case, they use atom transfer radical polymerization. The incorporation of the end groups is already here in the catalyst of the polymerization. Then the bromide that appear here, it was modified by acide. This is an example of unimolecular reaction. It's only one molecule with two different chemical groups. Then, well, there are many examples in the literature after this first example of the use of click chemistry, but I brought this one here because in this synthesis, they demonstrated that the use of strain-promoted acide alkyne click reaction, the use of metal catalysts can be avoided, which is probably good for biological applications, when we don't need to use a metal catalyst. This is an example of bimolecular reaction. This occurs also very fast, and there's many examples. For example, it's a table with plenty of examples of found from the literature where different kind of polymers, polystyrene, EPAN, butyl acrylates, etc., can be synthesized by different polymerization techniques using the copper catalyzed acide alkyne reaction can be generate these cyclic polymers. Diels-Alder reaction, very common in organic chemistry, as being also very useful in the synthesis of cyclic polymers by using heat. And after this first example, then UV light could be was used for the generation of these cyclic polymers. This work was done by Barnett Kobolik in 2014, and he implemented the use of this group, which by activation with light from this diene, and that reacted on the light conditions with the alkene group and close the chain and form the cyclic polymer. Many examples in the literature about the use of these and other reactions. I will not stop here because so many reactions, many words, but only to show you that there are many examples in the literature. This is another approach to generate cyclic polymers. Ring expansion polymerization is different from the ring closure in the sense that we can work in bulk conditions, concentrated solutions, we can obtain higher amount of cyclic polymers. The method is related to the specific monomer to catalyst interaction. We cannot synthesize all kinds of polymers with this approach. Initially, the monomer or the catalyst can be cyclic. And then, upon certain conditions, the monomer start to incorporate into this cyclic structure, and the ring is expanded, and form the cyclic polymer, something like this. These are the typical polymerization technique: ring expansion, metastasis polymerization, sweeter ionic, cationic, anionic radical polymerization. I brought some examples. This was the first example where a ruthenium catalyst was used for the generation of a cyclic polyolefin by Robert Grubbs in 2002. He demonstrated that this process is efficient, produce high amount of cyclic product, and allows us to synthesize a cyclic polyolefin that was not possible before. This is a kind of a ring expansion metastasis polymerization. This is an example of this. This is related to the incorporation of this monomer into the cyclic catalyst of ruthenium. Then this is the polyolefin that's obtained, and by hydrogenation, cyclic polyethylene can be obtained. So there are some modification of this ruthenium catalyst, for example, by modifying this linker here, and other monomers were also able to be polymerized and to obtain the cyclic structures, like for example this cyclic polynorbornene. This is a very recent work. As you see, over 20 years, there have been every time more and more examples and approaches to synthesize a nice cyclic polymer. The catalyst word is also very important here in the synthesis of cyclic polymers. These are methyl catalysts developed in the group of H. This is in Florida, in a university in Florida, where they have demonstrated that these catalysts, they just modify the linker here. In this case, they modify the methyl, and they are able to obtain cyclic polynorbornenes, polyacetylene, etc. These are the very important papers from the group, where they demonstrated the use of this, the development of this catalyst and the generation of the cyclic polymers. This is related to the sweeter ionic ring expansion polymerization. This is another technique to generate cyclic polymer, also by this ring expansion. This was demonstrated by Heydrich and Weymouth in 2007. They demonstrated that by using organic catalysts, no methyls, organic catalysts, this carbene was able to polymerize the lactide and to generate cyclic polylactide. This is a polyester, very important in industry, in the package, and because it's biodegradable and etc. He demonstrated in further experiments that this is probably a sweeter ionic mechanism, but in which the nucleophilic attack of this catalyst to the monomer create this growing chain with the two ions here, the sweeter ion here, negative and positive. They travel together, and then more monomer is incorporated into this structure, cyclic structure, and the ring expands upon certain conditions. The catalyst is released and the cyclic polymer is formed with no linker, only the monomer, same monomer. This has been very important in the literature because of the importance of these polyesters in industry, etc. And there are many research on this topic. This is a general mechanism where this sweeter ion is formed and generate the cyclic structures. There are many nucleophiles and monomers that have been proved with this mechanism. Well, in 2014, we came with this system. I look at the literature. I found this previous work of Weymouth, and I say, okay, we have obtained something similar, but with another kind of monomer, which is an epoxide. But this mechanism seems to be also sweeter ionic, but not nucleophilic, but electrophilic, because this catalyst attack the monomer by electrophilically. Found it looks for electrons fixed here, and then start to open the monomer and to generate this growing chain also by a sweeter ionic mechanism. And at the end, the catalyst is released and form this cyclic polyether. And we found that by changing these groups here, with these side groups, we can obtain the cyclic polymers with different functionalities. Let's go to the detection and identification. How do we know that we get cyclic polymer? It's very important for a chemist. Imagine, of course, these are AFM pictures, but it's not easy, and we cannot imagine all the cyclic polymers. Why? Because in this case, this is a copolymer, where the cyclic polymer is made by other polymer, and the comb is polystyrene, or the brushes are polystyrene. In this sense, the walls of this cyclic chain are become thicker, and can be analyzed by AFM. But normally, a polymer are not so thick and cannot be measured by AFM. And also the size should be perfect to be measured by AFM to see this hole that the tip go through these cycles and can go through the holes. It's very nice. So you can see also impurities, linear impurities, and in this case, these catenated cyclic polymers that all the physicists ask me about this in the talks, because this is very, very nice. But in this case, this is an impurity. To synthesize this kind of catenated cyclic polymers have to be synthesized with some specific reactions. Okay. GPC, this is another technique that we use to identify cyclic polymers, as I said before, because they show smaller hydrodynamic volume. They are smaller. This is an example of Jordan in some recent work we did. So this is a dashed line. This is a linear precursor. Then we obtain the cyclic polymer, which is here, is shifted. But as you can see, we have a shoulder here. This is very typically found in cyclic polymers when you use this ring closure approach. And this decreases with the decrease of the molecular weight, because the end groups find easier each other. Of course, we have to purify these samples. We did here preparative GPC, where we can separate this from this, and this is the result, the purified cycle. So this is very nice. And then we can measure this sample specific physical properties without the linear impurities. GPC also, but with a triplet detection. This is a multi-angle light scattering, refractive index, viscosity. Here is the typical shift in the GPC data after the formation of the cyclic polymer. And these are the viscosity data in solution. For the same, for the precursor and the cyclic polymer. So they show reduced intrinsic viscosity of and with a ratio which is is in agreement with the literature, is about the 0.8 or 0.7, I think was in this case. So this is a demonstration of also having obtained the cyclic polymer. Malditov mass spectrometry. This is a mass spectrometry that measures the mass of a single chain for n times the monomer. So we have one line. The next line is n plus one, like this. This is an example from Ali works, where we have here the the polyethylene glycol connected with this linker. This linker has a mass of 434 Dalton. And when we compare the linear precursor and the cyclic precursor, we observe the shift exactly by the same mass amount of the linker that we put in this cyclic. So this is another way to identify the cyclic polymer. And this is more recent. This is by using broadband dielectric spectroscopy. We implemented this to identify the formation of cyclic polymers. A linear, but this is true only in linear polymer. For example, a linear polymer where we have all the monomers oriented in the same direction. This means in chemistry work, a regular polymer. It should be a polymer that shows this, what is called normal mode relaxation. What is a normal mode relaxation? Is related to the fluctuations of the end-to-end vector of a linear chain. Not all the polymer shows that, but this polymer, and specifically this polyether, shows this end-to-end vector. Then this the dipole moment along the chain contour is proportional to the end-to-end vector. So we get a signal which is proportional to the molecular mass of the polymer. What happens if we cycle this polymer? The dipole moment is cancelled. So this normal mode relaxation should disappear, and disappear, in fact, here. This is the alpha relaxation, which is related to the glass transition. We use this as a comparison. All of them have it. And this is an impure sample, where we got some linear impurities. It shows a signal here. But our cyclic polymer has no signal. So in this sense, we could identify that we got the cyclic polymer. Potential applications. Well, applications are related to the physical properties of the polymers, of course. I find that this the application in the biomedical field is very interesting. Why? Because cyclic polymers have problems to go through pores. They diffuse with more troubles than the linear polymer. The linear polymer goes through only one chain, moving peptide through a pore. Cyclic polymer, two parts of the chain have to travel together through the pore. And this has been used and has been explained by what is called enhanced permeability and retention effect. Very typical use in the biomedical field. And in this first publication, in the Journal of American Chemical Society in 2009, the author demonstrated that the pharmacokinetics and biodistribution of cyclic polycaprolactone was different than in comparison with the linear polymer. So it was retained more in the body of mice. They used mice to demonstrate that. And with this, it's very useful, for example, to attack tumors, because in the cyclic polymers retain more in the tumor cells and can be used for medical purposes. What are the challenges here in the cyclic polymers? Topological purity, as I said before, we have many byproducts, large batch production. And when we get all this, then we could think that this cyclic polymer become commercial. So these are the main challenges in the cyclic polymer work. So I have talked about properties, synthesis, detection, potential application, cyclic polymer. Let's see what we do in our lab. So these are recent work with the cyclic polyethylene glycol that we use as nanoparticles surface ligands in collaboration with Marek. Polyethylene glycol is very much used in the stabilization of colloidal metal nanoparticles. And in this project, we think, okay, what happened if we use cyclic polyethylene glycol instead of linear polymers? Something is gonna change? Yes, things change it, as I will explain. First of all, we developed a strategy to synthesize the cyclic polymer that contain a thiol group, because the thiol group is important to attach to gold. In this case, we have to protect the thiol group, and we observed that the protection group for thiol was very convenient because we can leave it in the polymer, and it's good for storage and etc. Then we modify flat surfaces because we wanted to demonstrate if this reaction was possible. We demonstrate that by XPS and NMR that this group is released, that the polymer is attached by a thiol group, and then that the surfaces are modified with the cyclic, and we did the same with a linear equivalent. Okay, and we measure the contact angle of these gold surfaces modified with the cyclic polymer and the linear polymer, and we observe a decrease of the contact angles, meaning that the gold become more hydrophilic. That's good, but we observe also differences in the cyclic brushes comparison with the linear polymers. So the value here was a bit higher, well, 10 degrees higher than the linear one, meaning that this is more hydrophobic than the linear one. So we can modify surfaces with the cyclic polymers and make it more hydrophilic, more hydrophobic, depending on the modification. Then we went to the nanoparticles, and with the same experiments, we used the same polymer by ligand exchange protocol, and we observed differences in the colloidal properties with the temperature, with the presence of ions, etc. And if you look at the picture, you can imagine. So I draw here. So 5 chains, 5 and 5. But if you see here, the density of chain here near the surface is much higher than here, because two parts of the chain have to be connected to the gold. And this increase in the concentration of chains near the surface modify or have an implication on the properties, on the colloidal properties of these nanoparticles. This was the last experiments we did with this polymer. We put it in ethanol, the nanoparticles, we put it on the fridge, and we observed that in the fridge at minus 25 degrees, the linear brushes allow the precipitation of the nanoparticles, but the cyclic remain stable during months. Samples are still there, still stable. And then we did some experiments, and we established or explained the results by experiments, and with this mechanism by which by cooling the polymer collapse. Of course, it happened mostly on the polymers. They collapse because of the cooling. Then very slowly, this is a very fast process, very slowly they start to interpenetrate, and seems to be they're responsible for this precipitation. The cyclic polymer, this is not possible, and probably this is an explanation for that. I have explained very briefly. There are more details here, but I cannot explain all this. This is the last part. I will explain what we are doing in our lab. This is the most recent we are working with. Is on branched cyclic polyglycidol as a carrier of biological active molecules. This is in collaboration with Luca. As I said before, in 2014, we discovered this the formation of these cyclic polyethers by a pseudo ionic mechanism. Okay, we say, what happens if we change these phenyl groups by hydroxyl groups, and then to get a hydrophilic polymer, a water-soluble polymer. We did it, and things became more complicated. Why? Because the catalyst is able to react by the oxygen of the ethyl group, but also through the hydroxyl group. And then the polymer structure is more heterogeneous in the sense that there are more type of structures here, but also the branching reactions. This hydroxyl group can react with the growing chain and can form branches. The cyclic polymer are formed, as we identified by Mandy, but we also observe different features on the polymerization that also is possible that the ring fusion of two chains could happen that would explain the increase of the molecular weight of these systems. However, what we get is something depicted here as a branch cyclic polymer with a lot of branches full of hydroxyl groups, which resembles this hyperbranched polyglycidol, very much studied in the literature for biomedical applications because of the possibility of modifying these hydroxyl groups. And this is what we are doing. We are trying to use this cyclic polyglycidol, which is synthesized very easily just by putting the monomer, the catalyst, and we get the polymer. High amount of polymer is here. It's very viscous. We can fractionate this polymer and get different molecular weights. And we can do the modification of the group. This is the work that is doing Andrea right now with these cyclic polymers. He's attaching molecules that are synthesized in Lucas group. These are flavins, and that provide well, some properties to the polymer. They are activated by light, and they can promote some further reactions to the platinum. Also, platinum can be attached here, and we can obtain a cisplatin, for example, which is useful for in cancer treatment, for example. So we are working on this right now, and that's all. Thank you for your attention.
Thank you very much for a nice overview. Time for questions. Hello.
Hi, very nice talk. Thank you very much. Great. Quick question. I don't know. I guess that you know about the other types of polymer, well, cyclic sort of like polymers, like cyclic peptides, and they can sort of like stack between each other, forming like tubes, and they're used for membrane penetration and things like that. So I was wondering if you know if these polymers can actually stack and form like long tubes or like short tubes as cyclic peptides.
I haven't read about this from experimental work, but as I know, Angel Moreno, who is this guy, he modeled these polymer chains, cyclic polymer chain, and he observed the stacking of these polymer chains. He asked me to measure, see if we are able to determine that. I say, okay, from the experimental point of view, I see it very difficult in our polymers, but it seems that this is possible. They can stack small sizes, not big sizes, small sizes.
Yeah, of course. Cyclic peptides.
And it has to be stiff. Yeah. In cyclic peptide, I have no experience with cyclic peptides. I don't know. I don't.
They usually force I mean, there's a big field about it. They usually force the piling using changing the same residues.
Mm-hmm.
If you are aware of it, because I mean, you know that so that there are biological cyclic polymers that are not that they have knots in the structure. Mm-hmm. Yeah, exactly. For example, they are supposed to proteins with knots are supposed to be more stable, so against the naturalization. So is it possible to get not not synthetic polymers, and which kind of routes they follow? You know that?
No. They probably are formed in long chains, can form this kind of knots.
Like if they are confined, surely at some point then they will form it. But
synthetically, you have to think about how to do that. There is a Japanese group who did a very interesting work. he did a linear polymer, and he put like a polyrotaxan, I think it's a rotaxan or something like this, and he can move this cycle to the end of the line of the chain or to the other side, and he can create this kind of cyclic or linear as a to the convenience. And so you can do whatever you want with the synthesis, but you have to design very well these systems, and you have to demonstrate it, of course, and it's complicated.
What is the size of your rings?
The size.
The size of the rings.
For example, the brushes.
For example, the last one you mentioned with these examples of the Luca.
The radius of gyration is about 2 nanometers or something like this.
They are very small.
They are very small. Well, the molecular mass are still small.
And they are flat. What is that? They are flat?
No. I don't think they are flat, because they have a lot of branches and so on.
Yeah.
But the best measurement that we have are these systems, where we have the measurement of this. The height, the brush height is about depending on the molecular weight, the highest we got is 25 nanometers, something like this. The height of this brush with the cyclic polymer, with 10,000 molecular weight.
But in the last example you mentioned concerning, could you put the last slide? In this case, I realize I am not expert, but I am not expert in chemistry in these kind of things. But if you have a look at this plot and you see that the structure you are linking to the cycle is even larger than the cycle.
No, you have an N here, so
that means this is more larger.
Yeah, it's like yeah.
And what is the advantage, the real advantage? Because in many cases I'm not sure about what is the real advantage of all these kind of things related with the linear polymers. That means, for example, in one case like this, what is the advantage of having a cycle concerning having a small polymer with the same terminal or the same functional groups? Because
okay, we are our guess and our experiments are directed to this direction, is related with this property of this, okay? Because we are going to this direction, to the biomedical application of these polymers. They are water-soluble polymers, and we want to demonstrate that they behave in a different way, and probably in a better way than the linear chains, by coupling, attaching biological active molecules. In fact, we have just started collaboration with Milano Bicocca, where we are putting fluorescent molecules, and we want to study this in cells. So we are going into this direction, but just based on this picture for biological applications. The branches help also here, because with branches, this is more difficult to go through pores. So, but we still have to demonstrate that.
At the beginning of your talk, you've been showing these like different diagrams with like these theta or glass temperature, these heat capacity thermograms, and then I think there was these measurements of solubility or something like this, which I guess point at the single molecule or dilute behavior and the phase behavior of the polymers. I don't know if you're aware of this relationship that has been found between the theta temperature and the critical temperature for intrinsically conserved proteins. That is very nice because it connects the like single molecule properties with the phase behavior. I was wondering if anything like this has been done for circular and linear polymers to relate both things.
Related to proteins, you mean?
No, with just these more general like linear and cyclic polymers.
Related with the solution behavior.
Well, whether there is this connection, there is this fundamental relationship between single molecule properties and phase behavior.
And phase behavior. Well, you have examples of this, for example, thermoresponsive polymer, Nipan, not all the polymers have this behavior. There are also measurements of the theta temperature on some polymers. They have shown, I don't bring it here, the sample, but they have shown different theta temperatures for the linear and cyclic polymer or equivalent molecular weight. So there's some, there are experiments and there are results on this in direction too.
Before maybe we go to your question, let me read the question from Bo Chen.
Okay.
Not to discriminate our audience in online.
Yeah, okay.
So is there any difference in the solubility between cyclic and linear polymers?
Can be some differences, but it depends on the polymer, on the nature of the polymer. Yeah, there are some data also related to that, but not all the polymer. In my case, I have seen similar solubilities, but it's also related with the phase behavior and so on. Yeah.
So I have a curiosity about broadband dielectric spectroscopy and the comparison between linear and cyclic. If you can also show the graph, that is, so the alpha relaxation, we see that is shifted to lower frequency because we have some, the TG is higher also in calorimetric results. But I wonder if there are some other secondary relaxation like beta, gamma, and if there's some differences in.
Yes, the beta relaxation we also shows some differences. In this work, specifically, we centrated the attention in the normal mode relaxation because we synthesize this polymer on purpose. We have a previous work done, the master thesis of Thomas, where we synthesized, we analyze this polymer. This is a polyglycidol phenyl ether synthesized by this method. This is not true what I depicted here because I depicted the regular polymer. In this reaction, we don't get regularity. So the direction of the polymerization can occur through this carbon or this carbon, and this introduce irregularities. In that work, we analyze the beta relaxation of this polymer, and we observe that how they ended this beta relaxation could give us information about the presence of hydroxy groups at the end. In that, about the regularity, we couldn't use the normal mode relaxation because of the irregularities in this chain, but the beta relaxation was very good for identifying the formation of these cyclic chains.
So because it's interesting for me in calorimetry and in the vitrification of the system, because this can affect also the vitrification of the system. Maybe if we cool from the liquid polymer to a glass. Maybe it's interesting to see differences also between this head, tail, polymerization and the linear, if there's some differences.
The vitrification, so
yes,
glass transition.
Yes.
The
but
glass transition of
also
this is
the
higher.
relaxation of the glass. Below the glass transition temperature, we have some different kinetics, maybe, I don't know.
Okay, by calorimetry, we only measure the glass transition, that's it. But by the electron spectroscopy, we have a lot of measurements with this polymer. The regular, the regular. We also synthesize it with two arms. We close it. The normal mode did not disappear because we have two arms. There is a dipole moment resultant in this polymer. And we study the dynamics of this cyclic polymer by using the normal mode relaxation of this cycle. So with same polymers, this same polymer, I have, I don't know, several studies.
It could be a perspective, future perspective.
Yeah, we can talk.
Okay. Thank you.
If there is no more question, I have one very quick one. You show a different type of polymers, techniques, and so on. Is there any literature data on the cyclic water-soluble conjugated polymers?
Water-soluble conjugated polymers. Electronical conjugated, you mean? The conduct the electricity. Oh, no.
I don't think so, that's fine.
But there are some studies on conductive polymers, cyclic conductive polymers. This, of course, but I don't have it in my mind. And that they are water soluble, don't know.
Maybe one last curiosity. So it's clear that the synthesis is a major hurdle for studying these systems. Is there something to be learned from nature which has solved this problem for building like cyclic DNA? Is it because it uses a specific enzyme, or it's a templated process which we cannot do in the lab? Or is there something that we can learn from nature to emulate synthesis?
To be honest, I'm just reading about this because I'm writing a paper review, and I would like to include this part on how researchers are synthesizing cyclic DNA RNA by using templates, enzymes, and in fact, there are some reviews on this, and they are using it, these templates. Yeah, I know that it's done. So it's done in the lab. So how nature synthesizes these polymers, don't know. I don't know. Enzymatic processes, of course. The thing is that in the lab we can obtain a small molecular weights polymer or DNA or RNA because of problems with how they found each other, the end groups how they found each other, problem with the beta conformation. I don't know, the proteins, they start to, I don't know, to form beta sheets and kind of things, and then they cannot react as a flexible polymer, let's say.
Concerning the synthesis, the synthetic methods, the ring expansion procedure seems to be very simple and very fast, no? Do you know some industrial applications or something like this of these methods? Or?
It looks like the American group that is working on this catalyst. They are developing this catalyst for something. To get this polyacetylene, polynormal, they are all industrial polymers, important polymer. They are developing this area of the catalyst because this is the most important in this route. You need a catalyst for a specific monomer, and you get large amount of the polymer. So to go to the industrial applications, you need to go to the catalyst.
I'm not sure if you're showing, but what I remember is that what I recall is that all the cyclic polymers that you've been showing are basically made with a single monomer. So the question is, in like linear polymers, they can form like, well, statistical big block, 3 blocks, many different kind of things. Has anybody done something similar with cyclic polymers, just mixing and matching monomers, and how? Do you know if there is anything like
These
not cyclic.
these people mix monomers and they got a block cyclic polymers. It's possible to obtain it. Yeah, it's possible with this route. Yeah, they have done it. So you mean it was a block copolymers, or block or statistical copolymers?
Exactly. Basically.
Yeah, it's possible.
It's possible.
And in fact, I did it also. I also did it. I put here also THF.
Okay.
That was by serendipity because I was using THF as a solvent, and I saw that it copolymerized here. So I got a cyclic copolymer was this monomer and the THF.
And do they have like, so you have like a block or diblock cyclic polymer? Do they have like different properties compared with the linear, other than what you have from a cyclic polymer? Or?
My cells, you can create my cells with different properties.
Yeah, but that's something that you also can do with the linear. What I'm wondering is if there is something else that you can do.
Something different with the cyclic that you cannot do with the linear.
I don't know if the fact that the diblock polymers are basically cyclic provides them an extra functionality or an extra properties. It's a big question.
You can think of many properties. I don't know. I don't know how they crystallize or whatever.
Okay, no more questions. So let's thank Fabienne again for this.